Hello and welcome. In this lesson, we're going to learn about cell physiology. Cell physiology can be defined as the study of the functions of cell structures, which include organelles, the cytoplasm, and the cell membrane. You will recall some of these cell organelles include the mitochondrion, the function of the mitochondrion is to carry out respiration, during which energy is released and made available for the use of the cell. Another important organelle found in plant cells is a chloroplast, whose function is to carry out photosynthesis. Photosynthesis produces carbohydrates that is used by plants and other organisms during respiration to obtain energy. Now, these processes that occur in organelles and even in the cytoplasm require certain raw materials such as glucose, also require nutrients, and they release products. For example, the glucose produced during photosynthesis, which must be removed from the cell and transported to other parts of the body where they're going to be utilized. For example, in an animal cell that has a typical is a nucleus, oxygen moves into the cell and is used in the process of respiration, during which carbon dioxide is released. Glucose will also move into the cell to be used in the process of respiration. Therefore, it is very important that each living cell has an elaborate way by which substances get into and out of the cell. Movement of substances into and out of the cell is controlled by the cell membrane. So it is very important that the cell membrane is structured in such a way as to control what gets into the cell and out of the cell. Membrane. You will recall that the cell membrane consists of 3 layers. And these 3 layers are: 1, there is one phospholipid layer that is sandwiched between 2 protein layers. Now, the phospholipid molecule consists of the fatty acid part and a phosphate molecule. So this will be the fatty acid and the phosphate molecule. Actually, there are 2 layers that make up the phospholipid layer. Then on either side of it, there is the protein layer. Within the cell membrane, there are pores. The functions of these pores are very, very important. We're going to discuss this function shortly. Now, if you look at the cell membrane, there are certain features of it which gives it the properties that control the movement of substances into and out of the cell. These features that play this role include you have the pores. Then on either side there are electric charges, both positive and negative charges. Then by the very nature of this cell membrane, particularly the protein, the cell membrane is sensitive to changes in temperature and the pH of the surroundings. Then the thinness. The cell membrane is quite thin, and this has a great influence in the rate at which materials will be moving into and out of the cell. Then the structural proteins also have some additional role which influence the movement of materials into and out of the cell. We looked at the features of the cell membrane that enable it to control the movement of substances into and out of the cell. These features also give the cell membrane the properties which determine the passage of substances into and out of the cell. We look at these properties one by one and relate them to the movement of substances. The first will be the selective permeability, also known as semi-permeability. The tiny pores on the cell membrane will allow substances that are small enough through, but not those that are too big to pass through. So that's the cell membrane, and here we have the pore. Now this property enables the membrane to select what enters and leaves the cell. For example, sugar molecule like sucrose is too large to pass through, while water molecule, being small enough, will pass through the pores in the cell membrane. The membrane is therefore said to be semi-permeable. Semi-permeable, and this property allows the membrane to select what enters and what leaves the cell. For this reason, the cell membrane is said to be selectively permeable due to the presence of the pores that determine what size of the particles will pass through and those that will not be allowed to pass either into the cell or out of the cell. The second property of the cell membrane that influences the movement of substances in and out of the cell is the presence of electric charges. An electric charge can be positive or negative. In most cells, the outer membrane surface has a positive charge. So the positive charges are spread over the outer surface, while the inner surface, that is the inner surface of the cell membrane, has a negative charge. So the negative charges will be spread over the inner surface of the cell membrane. Now this type of membrane now is said to be polarized. They are opposite poles, whereby the outside has a positive charge relative to the inner surface. So the membrane is said to be polarized. Now the presence of these charges determine which substances will move in or out, and also at what rate. Charged particles would move faster across the cell membrane than the neutral non-charged particles. For example, a negatively charged particles would move faster across the cell membrane from the inside to the outside than a positively charged particle. This is because of the polarization of the cell membrane. This negative charge particle will be attracted by the positive charge on the outer surface, so it will move faster compared to the positively charged particle. On the other hand, a negatively charged particle will cross the cell membrane faster from the outside and into the cell, again because of the polarization of the cell membrane, whereby the outside is positive and the inside is negative. So the negative charge on the inner surface will attract the negatively charged particles that will then move faster than a neutral or positively charged particles into the cell membrane. And that is how electric charges affect the permeability of the cell membrane and the rate at which substances will move across. The third property of the cell membrane is its sensitivity to changes in temperature and pH. Now, the proteins in the membrane function best within a particular range of temperature and pH. Any change in the temperature and the pH will change the nature and thereby deform these proteins. High temperatures and extreme pH will denature the structure of the protein and therefore the membrane, resulting in loss of normal functions. Now, this is the normal cell membrane with the pores present that regulate the movement of substances in and out. Now, when exposed to high temperature or extreme pH, that is too much acidity or alkalinity, then the structure of these proteins will change. Some of these proteins will get deformed and the pores that are present are blocked such that substances cannot freely move through. Or because of the deformity, the pores become too wide and such that there is no control or regulation of the substances that move through, and those particles and molecules that previously could not pass through will move through freely, such that what results from the deformation and or denaturation of the cell membrane is loss of semi-permeability. And once that happens, then the cell membrane cannot function normally. Another property of the cell membrane is its thinness. Even though the membrane has 3 layers, it is very, very thin. In fact, it is approximately 7.5 nanometers in most cells, which is about 7.5 times 1/10 to power 9 meters. Quite thin. Now, the thinness promotes rapid movement of substances into and out of the cell. Even though the primary function of the cell membrane is to contain the materials within the cell, but it also controls what moves in and out. And it is important that it is thin enough to allow for the rapid movement of substances into and out of the cell. Now, the fifth property of the cell membrane is due to the presence of the proteins. Remember, the proteins primarily are structural components of the cell membrane, but in addition to that function, they also facilitate the movement of substances across. This occurs in 2 ways. First, by forming channels within the cell membrane through which the substances will pass. Now, at various points within the cell membrane, the proteins are arranged in such a way as to create channels or tunnels through which certain molecules can move either from the outside to the inside, or the reverse, from the inside of the cell to the outside. So without these channels, these molecules, which in many cases are normally too large to pass through the normal route across the cell membrane, thus they can only pass through the special channels that are created by the presence of the proteins within the cell membrane. Now, the second way in which these proteins facilitate the movement of substances across the cell membrane is by acting as carriers. So they're known as carrier proteins. The carrier proteins are located within the cell membrane. So in each cell within the cell membrane, there are particular proteins that move across, back and forth within the cell membrane, and in the process ferry certain molecules across. So the specific molecules will attach themselves onto the carrier protein, which then moves across from one surface to the other. Upon reaching the other surface, the molecule is released. So in this way, these proteins facilitate the movement of other molecules either from the outer surface into the inner surface, or vice versa, that is from the inner surface to the outer surface across the cell membrane, of substances into and out of the cell. Substances constantly move into and out of the cell for various reasons. Now, it is very important that materials constantly move into and out of the cell. So we're going to list the importance of this movement. Why do substances keep moving into and out of the cell? Now, depending on the nature of the substance, the movement will be for the following reasons. One, it enables the obtaining of nutrients. It facilitates the acquisitions of nutrients. For example, glucose is a very important nutrient that all cells require, because it is the substrate that is used in the process of respiration to release energy. So it is important that glucose is constantly supplied to a living cell. Another reason why substances move across the cell membrane is excretion. Now, excretion is the removal of the waste products of metabolic reactions. Now, these waste products cannot be allowed to accumulate because they will interfere with the normal functioning of the cell. In fact, these waste products are quite toxic, so they must be removed through the process of excretion across the cell membrane. The third reason is secretion. Cells produce useful cellular products such as enzymes, which function outside of the cell. So these enzymes must be released to the outside. For example, digestive enzymes must be released into the alimentary canal, where they will break down food substances into simple, soluble products. So the movement across the cell membrane facilitates the secretion process. Another reason for the movement of substances is for the attainment of ionic balance. in that there may be too many positively charged particles inside the cell. So it is very important that the excess particles are discharged, they are removed in order to attain the normal ionic balance within the cell. Now, there are three processes by which substances move into and out of the cells. So look at the processes by which these substances move into and out of the cell. So whether it is acquisition of nutrients, excretion, or secretion, or ionic balance, the substances will be moving through one of these three main processes. And the first one is diffusion, then osmosis, and 3, active transport. These are some of the main processes by which materials move into and out of the cell. There are other processes, but these three are the main ones that you're going to. Diffusion can be defined as the process by which particles move from a region of high concentration to a region of low concentration. For instance, from this region where they are highly concentrated, they diffuse to a region of low concentration. Is similar to particles moving down a gradient, that is from a region of high concentration to a region of low concentration along a concentration gradient. So diffusion is moving along a concentration gradient, that is from a region of high concentration to a region of low concentration. Now, how do you explain this process of diffusion? Explanation. Now, particles in a gas or liquid state are constantly moving freely, bumping into one another and bouncing off randomly. Now, because of this constant random movement, the particles tend to distribute themselves evenly throughout any space in which they are confined. So the higher the concentration at a particular spot, the greater the movement would be out of that particular spot and into a region where they are less concentrated. Now, the difference in concentration of particles between the two regions is known as a concentration gradient. Concentration gradient or diffusion gradient. And for as long as there is a diffusion gradient, particles will diffuse. Particles will diffuse from a region of high concentration to low concentration until the concentration in both regions become equal. That is an diffusion rate. Now, the rate at which substances move into and out of the cell is greatly influenced by a number of factors, which include: the first one is the diffusion gradient. Now, As you saw earlier, diffusion gradient is the difference in concentration between two different regions. So for instance, you can have two different diffusion gradients. Here there is this gradient, so let's call it this gradient A, and another one that is gradient B. Now, the greater the gradient, the higher the rate of diffusion. Gradient A is steeper and greater than gradient B. Hence, particles will diffuse at a faster rate from, say, region A to B than from region C to D, because of the difference in the gradient. So the higher the gradient, that is the difference between the concentration at A and B and C and D, the faster the rate of diffusion. So in this particular case, particles will be moving at a faster rate from region A to B than from region C to D. The second factor that affects the rate of diffusion is temperature. The higher the temperature, the higher the rate of diffusion, because an increase in temperature increases the kinetic energy of particles, thus making the particles to move faster from region of high concentration to region of low concentration. 3. Size of the particles. The smaller the particles or molecules, the higher or faster the rate of diffusion. Reason: small particles have lower weights, thus move at a faster speed. Just like by comparison, in motor vehicles, a small car moves at a higher speed than a truck because of its lower weight. Then number 4 is the surface area to volume ratio. Surface area to volume ratio, whereby the higher the surface area to volume ratio, the greater the rate of diffusion. Now we can illustrate this relationship using two cubes of different volumes. We have cube A, let's call this is A, of 1 by 1 by 1 centimeter dimension. So the surface area here is equal to 1 times 1 times 6, that gives you 6, and then the volume is 1 times 1 times 1, so this is 6, gives you 1. Hence, surface area to volume ratio is 6 to 1. Then compare this to a second cube, a larger cube of, say, dimensions 6 centimeters to 6 to 6. This gives us a surface area of 6 times 6 times 6, that gives you 216, and volume of 6 times 6 times 6, giving you 216. Hence the surface area to volume ratio is 216 to 216, which is equivalent to 1:1. So you see that the surface area to volume ratio of cube A is larger than that of cube B. Particles would diffuse into and out of this tube at a faster rate than in the case of B, because there's greater volume, greater distance to be covered by the diffusing particle where the volume is large. So relative to the surface area, here the volume is very big, and hence the rate of diffusion of particles into and out of a large body is going to be slower compared to that of a smaller body. Now, the fifth factor that affects the rate of diffusion is thickness of the membrane, or even thickness of the tissue. The thicker the membrane, the lower the rate of diffusion. Reason: when the membrane is thick, then the distance to be covered is greater. So the diffusing particles take a longer time to move across. Compare this to these two membranes, or a tissue like epidermis, where we have, let's say, the membrane X and membrane Y. A particle from this point diffusing across to the other point will take a longer time than the particle, another particle from this point in membrane Y diffusing across to the other side. All factors like temperature being constant, then the movement across Y is going to take a shorter time compared to the movement across X. Now, we look at the role of diffusion in living organisms. There are a number of processes in living organisms that depend on diffusion. We start by looking at these processes in plants. Such a process is absorption of mineral salt from the soil solution into the cells within the roots of the plant. This is a process that depends on diffusion. Secondly, gaseous exchange. Gaseous exchange in plants is solely through diffusion. So for instance, during the day, in the presence of light, carbon dioxide diffuses into the leaf, where it is used in the process of photosynthesis, while oxygen, which is a byproduct, is released and diffuses out of the plant, while at night, that is in the absence of light, there is no photosynthesis and only respiration takes place. Respiration requires oxygen, so oxygen diffuses from the atmosphere into the leaf through the stomata, while carbon dioxide, which is one of the waste products of respiration, is released from the leaf and diffuses out into the atmosphere. A third process in plants that depends on diffusion is excretion. Now, there are many gaseous waste products that are released from the plant into the surroundings through the process of diffusion. And 4, translocation. Diffusion plays some minor role in the movement of manufactured food substances from the leaves to the other parts of the plant. So diffusion is one of the processes by which manufactured food moves from the leaves where they're manufactured and they'll be in high concentration to the other parts of the plant where the levels of these food substances are lower. Now, in animals, there are two main processes that depend on the process of diffusion. The first one is absorption of digested food substances such as glucose and amino acids from the small intestines, and in particular the ileum, where these substances diffuse across the walls of the ileum into the blood to be transported to the rest of the body. The second process is gases exchange. In animals, oxygen diffuses into the body where it is used in respiration, and carbon dioxide, which is a waste product of the respiration process, diffuses out. Many animals have specialized gases exchange surfaces which are adapted structurally to enhance the process of diffusion. In this lesson, we look at osmosis. Osmosis is a process by which solvent molecules move from a region of their higher concentration to a region of a lower concentration across a semi-permeable membrane. Now, usually, osmosis is associated with the movement of water molecules, in that it refers to the movement of water molecules from a region of their higher concentration. Like in this example, solution A has a higher concentration of water molecules compared to solution B. And in this case, therefore, water molecules will move from solution A, where they are highly concentrated, across to solution B, where they have a lower concentration, across this semi-permeable membrane. Now, this semi-permeable membrane can allow water molecules to pass across, but not the other molecules. In this particular case, the sucrose molecules are too big to pass across. Because of this semi-permeability of the membrane, only water molecules will diffuse across. Osmosis is therefore a special type of diffusion because it's only the water molecules that are diffusing across and not any other molecule. So sugar molecules, in this particular case the sucrose, are too large and they are restricted by the semi-permeable membrane and hence cannot move across. So in a nutshell, osmosis refers to the movement of water molecules from a region of their higher concentration, is solution A, to a region of their lower concentration, that is solution B, across a semi-permeable membrane. Now we can demonstrate osmosis experimentally using a Vesican tubing. Now a Vesican tubing, also known as a dialysis tubing, is a synthetic semi-permeable membrane that is used in separating small molecules from large ones in a solution. Now in this experimental setup, a Vesican tubing is half filled with sucrose solution. The Vesican tubing is supported through a string on a glass rod that is then immersed in a beaker that contains distilled water. Now, after some time, say about 30 minutes, it is observed that the Vesican tubing enlarges in size. Now, how do you explain that? The distilled water has a high concentration of water molecules compared to the sucrose solution. So the water molecules move by osmosis into the sucrose solution across the semi-permeable viscous tubing membrane. So as the water molecules get into the viscous tubing, they cause the enlargement, because remember, water molecules are particulate matter that occupy space and have volume. So as more and more water molecules get in, the viscous tubing enlarges. This enlargement is as a result of the influx of water molecules that move by osmosis from the distilled water into the sucrose solution. In this second experiment, viscous tubing that is filled with distilled water is placed in a beaker that contains sucrose solution. After about 30 minutes, it is observed that the viscous tubing reduces in size. It becomes smaller. Why? What is happening is that the distilled water in the viscous tubing has a high concentration of water molecules, which then move out by osmosis into the sucrose solution with a lower concentration of water molecules. As a result, the viscous tubing reduces in size because it loses water, and as the water flows out, it becomes smaller and smaller, and in fact it is said to be flaccid because of that loss of turgidity as a result of the outward movement of water molecules. Solutions that have different concentrations are separated by a semi-permeable membrane. Water molecules will flow from its region of high concentration to region of low concentration. In this illustration, there are two solutions, solution A and solution B, separated by a semi-permeable membrane. Solution A has a higher concentration of water molecules compared to solution B. Therefore, water molecules will flow from where they are highly concentrated in solution A across the semi-permeable membrane to solution B. Now, the flow of water molecules will continue until an equilibrium is established between the two solutions. That is, the levels of water molecules in the two will be equal. Now, if you look at solution A, there are fewer number of sucrose molecules. The sucrose molecules are the solids. So solution A can be said to have low solute concentration. Well, relative to solution B is said to have a higher solute concentration. Now, in terms of solute concentration, solution A has a lower concentration of solute, and in this particular case, the sucrose molecules, relative to solution B that has a higher concentration of solids. Solution A is therefore said to be hypo for below, and then tonic solute concentration, while solution B is said to be hyper for above, and tonic for solid concentration. We can therefore say as a general rule that whenever two solutions of different concentrations, that is tonicity, are separated by a semi-permeable membrane, then the direction of osmosis will always be from the hypotonic solution across the semi-permeable membrane to the hypertonic solution, and this will continue until the two solutions become equal in terms of water molecules concentration. Remember, the solute molecules like sucrose are too big to move across the semi-permeable membrane, so it is the water molecules that are small enough to pass across, and therefore they're the ones that bring the equilibrium in terms of water concentration. Let us look at what happens when two solutions of the same solid concentration are separated by a semi-permeable membrane. If you look at solution C, it has the same number of solute molecules as the water molecules concentration. So this makes this solution to be 50% sucrose solution. Likewise, if you look at the number of molecules of the sucrose and water in solution D, you see that they are the same. It is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 for water, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 for sucrose. So likewise, solution D is 50% sucrose. Now, these two solutions having the same concentration are said to be isotonic. Solution C and D are isotonic. So what happens in terms of movement of water molecules? There's an equal number of movement from one side to the other. That is, the same number of molecules will move from solution C to D. And likewise, you're also going to have the same number moving from D to C, so that there will be no net gain or loss of water molecules by any of the two solutions. That is, C and D will not gain or lose water. Solutions depends on the difference between the osmotic pressure. Now, what is osmotic pressure? Osmotic pressure of a solution can be defined as the tendency of a solution to take in water from another solution. That is, the ability of a solution to attract water molecules from another solution separated from it by a semi-permeable membrane. That is what osmotic pressure is. Now, why is it known as an osmotic pressure? Now, this can be demonstrated in an experiment using a U-tube that is filled with distilled water on one arm, and the other arm contains sucrose solution. The two liquids are separated by a semi-permeable membrane. Now, distilled water has more water molecules compared to the sucrose solution. So we're going to have osmosis taking place from the hypertonic distilled water to the hypertonic sucrose solution. As water molecules get to the wing or the arm of the U-tube with the sucrose solution, a pressure develops because the water molecules come in with some force. This force will cause the solution in the wing of the U-tube that contains sucrose solution to rise to a new level. So this rise in the solution is caused by a pressure generated by the water molecules as they move from the region of low solid concentration or high water molecule concentration across the semi-permeable membrane to the sucrose solution. So as the water molecules get in, they generate a force or a pressure that causes the solution to rise to a new level. Now, because there is a force generated by the influx of water molecules, that force is referred to as the osmotic pressure. And the more concentrated in terms of solutes a solution is, the greater its tendency to take in water from surrounding solutions by osmosis. So in other words, the higher the solute concentration, the higher the osmotic pressure over set osmotic pressure using a Vissking tubing, in which the Vissking tubing is filled with the concentrated sucrose solution, and then on the upper end of the Vissking tubing it is tied, and a long capillary tube is immersed into the sucrose solution, then tied tightly. The Vissking tubing is then immersed in distilled water. Now, after some time, it is observed that the level of the sucrose solution rises up the capillary tube to a new level. Now, why? This is because water molecules move from the distilled water by osmosis and into the Vissking tubing. As the water molecules move in by osmosis, the volume of the Vissking tubing increases, and this generates a pressure that acts upward and causes the solution level to rise. So it is osmotic pressure that is caused by the influx of water molecules as they flow into the Vissking tubing by osmosis that causes the level of the sucrose solution to rise up the capillary tube. Is the capacity of a solution to take in water when separated from another one by a semi-permeable membrane. Osmotic potential can also be viewed as the hidden osmotic pressure of a solution that would only develop if water was allowed to enter it through a semi-permeable membrane. So a solution, like for example this sucrose solution contained in a Vissking tubing that is not separated from another solution or water by this semi-permeable membrane, has an osmotic potential. It's not osmotic pressure, it is a capacity, the hidden osmotic pressure. It has the capacity to develop an osmotic pressure if it were to be separated from distilled water by this semi-permeable Vissking membrane. So a solution that is in a container like a beaker and is not separated from another solution by a semi-permeable membrane is said to have an osmotic potential. Now, if you are to immerse this solution in a beaker that contains distilled water, then through osmosis, water molecules will move into the sucrose solution in the Vissking tubing and increase the volume and generate a force. That force is osmotic pressure that will have been generated by the sucrose solution. But for as long as this solution is not separated from the distilled water by the semi-permeable Vissking tubing membrane, then we talk about the osmotic potential. So osmotic potential refers to the capacity of a solution to develop an osmotic pressure. The moment that solution is separated from another solution by a semi-permeable membrane, that is, the moment the solution comes into close proximity with another solution and between them there is only a semi-permeable membrane, then that solution will then develop osmotic pressure as a result of the influx of water molecules into this lesson. In this lesson, we are going to look at the factors affecting the rate of osmosis. There are two main factors that affect the rate at which movement of water molecules take place, and these two factors are osmotic pressure and temperature. We start by looking at the osmotic pressure gradient. Osmotic pressure gradient refers to the difference in the osmotic pressure between two solutions that are separated by a semi-permeable membrane. You will recall that the osmotic pressure is the tendency of a solution to take in water from another solution. The osmotic pressure is determined by the solid concentration, whereby the higher the solid concentration, the higher the osmotic pressure. Now take for instance these two solutions here. We have solution A and solution B. Solution A is 30% sugar, while B is 60% sugar. In terms of solid concentration, solution B has a higher solid concentration than solution A. And for that reason, solution B will develop a higher osmotic pressure compared to solution A. And as you'll remember, osmosis always takes place from a region of high water concentration or low solid concentration to a region of high solid concentration. In this particular case, osmosis will take place from solution A, where there is a high water concentration, to solution B. Now compare this with this other pair of solutions, of solution C and D. Solution C is 50% sugar, while solution D is 70% sugar. Now osmosis will take place from a region of high water concentration to a region of lower water concentration, or from a region of lower solid concentration, that is 50% sugar, to a region of higher solid concentration, that is 70% sugar. So water molecules will move by osmosis from solution C to solution D across this semi-permeable membrane. Now, the difference in osmotic pressure between the two solutions is what forms a gradient. In the first case here, there's a gradient or a difference of 30%, while in the second, there is a difference of 20%. This gradient determines the rate at which osmosis will take place. Now, in this case, the rate of osmosis is going to be higher from solution A to B than from solution C to D, because of a higher osmotic pressure gradient between solution A and B. So this way, the osmotic pressure gradient between two solutions determine the rate at which osmosis will take place. The second factor that affects the rate of osmosis is temperature. The higher the temperature, up to a certain point, the faster the rate of osmosis. Now why is this? It's because high temperature tend to increase the kinetic energy of water molecules, so they move at a faster rate, thereby bringing about a faster rate of osmosis. But an increase in temperature is only will only have a productive or a positive effect up to a certain point. High temperature will denature and destroy the cell membrane to a point that the membrane will be incapable of carrying out osmosis. So an increase in temperature will only apply up to a certain point, such that as the temperature increases, the rate of osmosis will also increase. But beyond a certain point, high temperatures will denature and destroy the cell membrane, thereby breaking the osmosis. It's a very important role in the lives of organisms. Movement of water is described as osmosis. So in this lesson, we're going to look at the exact role that osmosis plays in living organisms, starting with plants. One role of osmosis in plant is the absorption of water from the soil by roots. The only point of entry of water into the plant body is through the roots in most plants. And this comes about mainly through the root hair cells. The root hair cells have a protection known as the root hair that provides a large surface area over which osmosis does take place. So in the soil, water moves from the soil solution, which is hypertonic, and into the root hair cells by this process of osmosis. So in this way, plants are able to absorb water from the soil and get it into their body. Remember, the root hair cells have solutes in the sub-vacuole that raise osmotic pressure, such that the osmotic pressure of the root hair cells is higher than that of the surrounding soil solution. This is what facilitates the absorption of water from the soil solution into the root hair cells. The second role of osmosis in plant is in the movement of water from one cell to another. Once water gets into one cell by osmosis, it dilutes the sub-vacuole and the cytoplasm of that cell. So that cell becomes hypotonic relative to the adjacent cell. This facilitates movement of water from that cell into the next one by osmosis. The next cell becomes hypotonic in relation to the next cell, and the process goes on and on from one cell to another, and in this way water can move from one part of the plant to another part. For example, from the root epidermis into the cortical cells and all the way into the center of the root. Third role of osmosis in plants is support. Now, movement of water in the plant cells make them to be firm and rigid. The cells become firm and rigid when they become turgid. The turgid cells then provide support in the leaves and stems, especially of herbaceous plants. These are non-woody plants that do not have strengthening woody material, so they rely on the turgidity of the cells for support, and this turgidity comes about as a result of the inflow of water into the cells by the process of osmosis. Fourth role is in certain types of plants. These are the insectivorous plants. These plants grow in soils that are deficient in nutrients, especially nitrogen. So these plants must obtain the nutrients from another source apart from the soil, and they do so by trapping insects on their leaves, digesting them and absorbing the nutrients. This trapping comes about through sudden loss of water from the cells in the leaves by osmosis, leading to the closure. Now remember, one function of the turgidity of the cells provides support to keep the leaves open. Now, with the sudden loss of water, that support is withdrawn and the leaves shut, in the process trapping the insects within. These insects are then digested and the products and the nutrients absorbed into the plant. Another role of osmosis is in the opening and closing of stomata.
When water moves into the gut cells by osmosis, the gut cells become turgid. And when they become turgid, so this is a process, when they become turgid, the gut cells bulge outward, thereby opening the stoma. So when these gut cells are flaccid, the stoma is closed because the two gut cells are close together, they lie side by side. But as they absorb water through osmosis, the gut cells become turgid and they tend to bulge outward because the inner lining has a thicker cell wall compared to the outer lining. So it's much easier for the cells to bulge outward, away from one another, and in the process they create the stoma between them. So it is through osmosis that the opening of the stoma takes place, and also it is through osmosis when these turgid gut cells lose water, they become flaccid and come to lie close to one another, thereby closing the stoma. So opening and closing of the stoma depends on osmosis. Opening comes about when the gut cells gain water and become turgid, and closure comes about when the gut cells lose water to the surrounding epidermal cells, when they lose water and they become flaccid, lying side by side, and in that state they narrow or close the gap between them and the stoma is said to be closed. So that is those are the roles that osmosis plays in plants. When it comes to animals, there are two main roles that osmosis plays. The first one is absorption of water in the large intestines. In particular, the colon part of the large intestines is structured so as to enhance absorption of water from the lumen and into the surrounding blood capillaries. The second role is reabsorption of water in the kidney tubules. Most of the water that is filtered out of the blood is reabsorbed by osmosis into the surrounding blood capillaries. In this way, water is conserved in the body and not lost from the body through the urine. So the reabsorption process that takes place in the kidney tubules is by osmosis, just like also the absorption process that takes place in the large intestines also comes about through osmosis. In this lesson, we're going to look at water relations in animal cells with regards to osmosis. Animal cells have only a cell membrane enclosing the cell. This cell membrane is semi-permeable. Now, you'll also recall that animal cells lack subvacuoles, and that it is the cytoplasm that contains dissolved salts and sugars, and therefore the cytoplasm plays a very important role in osmosis as it determines the osmotic pressure within the cell. When an animal cell is placed in a solution, it will either gain water by osmosis or lose water by osmosis. It all depends on the osmotic pressure gradient. So if it is placed in a hypertonic solution, it will lose water by osmosis. And when placed in hypotonic solution, it will gain water by osmosis. Now, red blood cells are commonly used to demonstrate osmosis in animal cells. So we're going to look at how red blood cells behave when placed in solutions of different concentrations. We're going to start by looking at the behavior of the red blood cells when placed in hypertonic solution or distilled water. Now, hypertonic solution means that the solution has a higher water concentration compared to the cytoplasm within the red blood cells. Now, when a red blood cell is placed in such a hypertonic solution, the red blood cell will be hypertonic. So the red blood cell is said to be hypertonic to the surrounding solution. This results in a net gain of water. So water moves into the cell by osmosis. As more and more water enters the cell tends to swell. And if this continues, get more and more water entering the cell, it will swell further and eventually burst. This is because the cell membrane is not strong enough to resist the outward pressure that is caused by the inflow of water. Because remember, as the water flows in, because water being matter, it occupies space. So it will create an outward pressure which the cell membrane cannot withstand. And so eventually this leads to the bursting of the red blood cells, thereby releasing its contents to the surroundings. That is how the red blood cell behaves when placed in hypotonic or distilled water environment. Now, on the other hand, when the red blood cell is placed in a hypertonic solution, meaning a solution that has a lower water concentration relative to that within the red blood cell. Now, consequently, the red blood cell will be hypotonic to the surrounding hypertonic solution. This would therefore lead to a net loss of water from the cell by osmosis. Because remember, in osmosis, water moves from a region of its higher concentration, that is the hypotonic solution, to a region of its lower concentration, and that will be the hypertonic solution, across the semi-permeable membrane that is created by the cell membrane. So in the hypertonic solution, red blood cell will lose water, and as it loses water, it tends to shrink. It's going to become smaller because there's a decrease in volume. There's a decrease in volume due to the loss of water from the cell to the surrounding. Now, as the loss continues, the cell would shrink further and would appear wrinkled. Now, the shrinking of a red blood cell after losing water by osmosis is known as crenation. The process is known as crenation. And a red blood cell in the state of being wrinkled is said to be crenated. The red blood cell is said to be crenated as a result of the loss of water by osmosis. What actually happens is that the cell membrane collapses, because remember, the cell membrane is not strong enough to maintain the shape of the cell. So the loss of water leads to the crenation of the red blood cell. Now remember, in the body of animal cells, that is within the body of animals, cells rarely become hemolyzed or crenated. Why? Because the concentration of the body fluids and the blood is kept isotonic. That is, the body fluids and that of the blood concentration is kept isotonic to the cytoplasm of the cells by a process that is known as osmoregulation. So through osmoregulation, the bodies of animals, cells are prevented from crenating and from becoming hemolyzed. Osmoregulation is a very important process in living organisms because it ensures that the cells have the optimum environment in as far as osmotic pressure is concerned so that they can function at this lesson. In this lesson, we're going to look at water relations in animal cells with regards to osmosis. Animal cells have only a cell membrane enclosing the cell. This cell membrane is semi-permeable. Now, you'll also recall that animal cells lack subvacuoles and that it is the cytoplasm that contains dissolved salts and sugars, and therefore the cytoplasm plays a very important role in osmosis as it determines the osmotic pressure within the cell. When an animal cell is placed in a solution, it will either gain water by osmosis or lose water by osmosis. It all depends on the osmotic pressure gradient. So if it is placed in a hypertonic solution, it will lose water by osmosis. And when placed in hypotonic solution, it will gain water by osmosis. Now, red blood cells are commonly used to demonstrate osmosis in animal cells. So we're going to look at how red blood cells behave when placed in solutions of different concentrations. We're going to start by looking at the behavior of the red blood cells when placed in hypotonic solution or distilled water. Now, hypotonic solution means that the solution has a higher water concentration compared to the cytoplasm within the red blood cells. Now, when a red blood cell is placed in such a hypotonic solution, the red blood cell will be hypertonic. So the red blood cell is said to be hypertonic to the surrounding solution. This results in a net gain of water. So water moves into the cell by osmosis. As more and more water enters the cell tends to swell. And if this continues, we get more and more water entering the cell, it will swell further and eventually burst. This is because the cell membrane is not strong enough to resist the outward pressure that is caused by the inflow of water. Because remember, as the water flows in, because water being matter, it occupies space. So it will create an outward pressure which the cell membrane cannot withstand. And so eventually this leads to the bursting of the red blood cells, thereby releasing its contents to the surroundings. That is how the red blood cell behaves when placed in hypotonic or distilled water environment. Now, on the other hand, when the red blood cell is placed in a hypertonic solution, meaning a solution that has a lower water concentration relative to that within the red blood cell. Now, consequently, the red blood cell will be hypotonic. to the surrounding hypertonic solution. This would therefore lead to a net loss of water from the cell by osmosis, because remember, in osmosis, water moves from a region of its higher concentration, that is the hypotonic solution, to a region of its lower concentration, and that will be the hypertonic solution, across the semi-permeable membrane that is created by the cell membrane. So in the hypertonic solution, red blood cell will lose water, and as it loses water, it tends to shrink. It's going to become smaller because there's a decrease in volume. There's a decrease in volume due to the loss of water from the cell to the surrounding. Now, as the loss continues, the cell would shrink further and would appear wrinkled. Now, the shrinking of a red blood cell after losing water by osmosis is known as crenation. The process is known as crenation. And a red blood cell in the state of being wrinkled is said to be crenated. The red blood cell is said to be crenated as a result of the loss of water by osmosis. What actually happens is that the cell membrane collapses, because remember, the cell membrane is not strong enough to maintain the shape of the cell. So the loss of water leads to the crenation of the red blood cell. Now remember, in the body of animal cells, that is within the body of animals, cells rarely become hemolyzed or crenated. Why? Because the concentration of the body fluids and the blood is kept isotonic. That is, the body fluids and that of the blood concentration is kept isotonic to the cytoplasm of the cells by a process that is known as osmoregulation. So through osmoregulation, in the bodies of animals, cells are prevented from crenating and from becoming hemolyzed. Osmoregulation is a very important process in living organisms because it ensures that the cells have the optimum environment in as far as osmotic pressure is concerned, so that they can function at the water relations in plant cells. We first review the structure of a plant cell. A plant cell consists of a cell wall. Now this cell wall is made up of cellulose. The cellulose makes the cell wall rigid, strong and resistant to stretching. The cell wall is also fully permeable to materials. That means that most substances would freely move across the cell wall without any resistance. Now, this unique feature, that is the fully permeability of the cell wall, allows plant cells to survive in a wide range of osmotic pressure. Beneath the cell wall, there is the cell membrane, also known as the plasma membrane, that is semi-permeable and controls what enters and leaves the cell. Most plant cells have a large, usually centrally placed subvacuole. Subvacuole contains solutes dissolved in water. The membrane of the subvacuole is known as the tonoplast. It is also semi-permeable. It controls what enters and leaves the subvacuole. Then of course there is the cytoplasm, within which cellular organelles are suspended, including
the nucleus, the water relations in plant cells, we first review the structure of a plant cell. A plant cell consists of a cell wall. Now this cell wall is made up of cellulose. The cellulose makes the cell wall rigid, strong and resistant to stretching. The cell wall is also fully permeable to materials. That means that most substances would freely move across the cell wall without any resistance. Now, this unique feature, that is the fully permeability of the cell wall, allows plant cells to survive in a wide range of osmotic pressure. Beneath the cell wall, there is the cell membrane, also known as the plasma membrane, that is semi-permeable and controls what enters and leaves the cell. Most plant cells have a large, usually centrally placed subvacuole. Subvacuole contains solutes dissolved in water. The membrane of the subvacuole is known as the tonoplast. It is also semi-permeable. It controls what enters and leaves the subvacuole. Then of course there is the cytoplasm, within which cellular organelles are suspended, including the nucleus. This is the process by which plant cells lose water, shrink, and become flaccid, with the cell membrane detaching from the cell wall. If a plant cell is placed in a hypertonic solution, that is the one that has a higher solute concentration, more than the cytoplasm and the subvacuole concentration, then water molecules will move from the cell by osmosis into the hypertonic solution. And as this goes on, the cytoplasm together with the subvacuole will reduce in volume. If the loss of water continues, the cytoplasm and the subvacuole will continue reducing in volume to the point that the cell membrane becomes detached from the cell wall. Now, such a cell is said to be flaccid, one that has lost its turgidity. Remember, turgidity is achieved when the turgor pressure equals wall pressure. But as a result of plasmolysis, there is loss of turgidity, and with that loss of turgidity, the cell becomes flaccid. So you talk about a flaccid cell, one that has lost its turgidity. Now, as the shrinkage of the cell continues and the plasma membrane detaches from the cell wall, the cell becomes flaccid, or it's also said to be plasmolized. The cell is said to be plasmolized when it loses its turgidity. Now, when the water loss continues, you have more and more water being lost by osmosis. Total plasmolysis is achieved, and this occurs when the cytoplasm shrinks so much such that it just forms a mass, a spherical mass at the center of the cell. And the space between the cell wall and the cell membrane will be occupied by the solution, that is the hypertonic solution in which the cell is immersed. So all of this will consist of the hypertonic solution, because remember, the cell wall is freely permeable. So the molecules in the hypertonic solution will freely pass across the cell wall and get into the space between the cell membrane and the cell wall. Plasmolysis is the process by which a flaccid cell, when placed in distilled water or hypotonic solution, absorb water by osmosis and become turgid. In fact, it is the opposite of plasmolysis. Remember we said in plasmolysis that the cell, when placed in hypertonic solution, loses water by osmosis, and the cytoplasm together with the subvacuole shrink and the cell becomes flaccid. But in the case of deplasmolysis, the cell, when placed in hypertonic solution or distilled water, gains water by osmosis. The cytoplasm together with the subvolume will increase in volume, and the entire cell in fact increases in size. The cytoplasm together with the subvacuole exert an external or an outward force that presses against the cell wall, and the cell wall, being rigid, resists the stretching by exerting the wall pressure. So when the turgor, the outward turgor pressure as a result of the expanding cytoplasm and the subvacuole equals the opposite wall pressure, then the cell is said to be turgid. So deplasmolysis is a process in which a flaccid cell, starting with a flaccid cell, is placed in a hypotonic solution. The cell gains water by osmosis. The cytoplasm together with the subvacuole enlarge as a result of the influx of water and push against the cell wall. Remember, the outward pressure of the cytoplasm and the subvacuole is known as the turgor pressure. The turgor pressure pushes against the cell wall, which resists the stretching, and the resistance to stretching, the opposite and equal force exerted by the cell wall, is known as the wall pressure. So at a certain point, the wall pressure and the turgor pressure become equal, and that cell is said to be turgid. So deplasmolysis is a process in which a flaccid cell placed in hypotonic solution gains water by osmosis to become turgid. So simply it's the transformation of the cell from the flaccid state to the turgid state as a result of the inflow of water molecules by osmosis. Deplasmolysis is the process by which a flaccid cell, when placed in distilled water or hypotonic solution, absorb water by osmosis and become turgid. In fact, it is the opposite of plasmolysis. Remember we said in plasmolysis that the cell, when placed in hypertonic solution, loses water by osmosis, and the cytoplasm together with the subvacuole shrink and the cell becomes flaccid. But in the case of deplasmolysis, the cell, when placed in hypertonic solution or distilled water gains water by osmosis. The cytoplasm together with the subvolume will increase in volume, and the entire cell in fact increases in size. The cytoplasm together with the subvacuole exert an external or an outward force that presses against the cell wall, and the cell wall, being rigid, resists the stretching by exerting the wall pressure. So when the outward turgor pressure as a result of the expanding cytoplasm and the subvacuole equals the opposite wall pressure, then the cell is said to be turgid. So the plasmolysis is a process in which a flaccid cell, starting with a flaccid cell, is placed in a hypertonic solution. The cell gains water by osmosis. The cytoplasm together with the subvacuole enlarge as a result of the influx of water and push against the cell wall. Remember, the outward pressure of the cytoplasm and the subvacuole is known as the turgor pressure. The turgor pressure pushes against the cell wall, which resists the stretching, and the resistance to stretching, the opposite and equal force exerted by the cell wall, is known as the wall pressure. So at a certain point, the wall pressure and the turgor pressure become equal, and that cell is said to be turgid. So the plasmolysis is a process in which a flaccid cell placed in hypertonic solution gains water by osmosis to become turgid. So simply it's the transformation of the cell from the flaccid state to the turgid state as a result of the inflow of water molecules by osmosis. It is a process where plant leaves and the young stems shrink and droop during hot, dry weather. This comes about when the plant loses water to the atmosphere at a faster rate than it can replace through absorption from the soil. This occurs in herbaceous plants such as the one drawn in this diagram here, a potted plant. When it is exposed to hot and dry weather, it loses a lot more water than it can replace through absorption in the roots. Now, when plant cells are turgid, they provide support to the tissues. Like in this first instance, the leaves are well spread out, the stem is upright because of the turgidity of the plant cells. But when the plant loses more water than it can replace, the cells become flaccid. And when the cells become flaccid, the tissues and the organs lose their support, and what results is the drooping of the stem, and leaves go limp and fold, and this plant is said to be wilted. Now, as the leaves droop, there's less surface area that is exposed to the sun, and this lowers the rate of transpiration or water loss from the plant. So in time, the drooping and the falling of the leaves helps to minimize or reduce the rate of water loss through transpiration by the plant. And in many plants, drooping occurs in the heat of the day when the temperatures are very high, and therefore the rate of transpiration is high. But toward evening, after the plants have wilted and drastically reduce the rate of water loss, the plants tend to replace the amount of water that is lost, because during this time the rate of absorption from the soil is higher than the rate of water loss to the atmosphere because of the rolled up leaves and the drooped stem. So towards the evening, plants do recover from their wilted state and are able to stand upright, that is during the evening, and the leaves are spread out. It is a process where plant leaves and the young stems shrink and droop during hot, dry weather. This comes about when the plant loses water to the atmosphere at a faster rate than it can replace through absorption from the soil. This occurs in herbaceous plants such as the one drawn in this diagram here, a potted plant. When it is exposed to hot and dry weather, it loses a lot more water than it can replace through absorption in the roots. Now, when plant cells are turgid, they provide support to the tissues. Like in this first instance, the leaves are well spread out, the stem is upright because of the turgidity of the plant cells. But when the plant loses more water than it can replace, the cells become flaccid. And when the cells become flaccid, the tissues and the organs lose their support, and what results is the drooping of the stem, and leaves go limp and fold, and this plant is said to be wilted. Now, as the leaves droop, there's less surface area that is exposed to the sun, and this lowers the rate of transpiration or water loss from the plant. So in time, the drooping and the folding of the leaves helps to minimize or reduce the rate of water loss through transpiration by the plant. And in many plants, drooping occurs in the heat of the day when the temperatures are very high, and therefore the rate of transpiration is high. But toward evening, after the plants have wilted and drastically reduce the rate of water loss, the plants tend to replace the amount of water that is lost, because during this time the rate of absorption from the soil is higher than the rate of water loss to the atmosphere because of the rolled up leaves and the drooped stem. So towards the evening, plants do recover from their wilted state and are able to stand upright, that is during the evening, and the leaves are spread out. The cell is placed in distilled water or a very dilute solution, that is one that is a hypertonic. Then water molecules will enter into the cell by osmosis and move into the cytoplasm and the subvacuole. As more and more water enters the cell by osmosis, the volumes of the cytoplasm and the subvacuole will increase, causing the cell to swell. So the cell swells as a result of the influx of water molecules, thereby expanding right from the subvacuole, the cytoplasm, and outward. The contents of the cell, that is the cytoplasm and the subvacuole, exert an outward pressure that presses against the cell wall. This outward pressure is known as the turgor pressure. The turgor pressure presses against the cell wall. Now, remember, the cell wall is rigid as a result of the cellulose. So in resisting the stretching, the cell wall will exert an opposite and equal force. This opposite and equal force exerted by the cell wall is known as the wall pressure. So as the target pressure increases, the wall pressure also increases, because target pressure pressing against the cell wall is one that creates the opposite wall pressure. Now, as more and more water enters the cell, the target pressure will increase, and as a reaction, the wall pressure will also increase. So the cell will continue to expand, but remember, a point will be reached when this inelastic cell wall will resist further stretching. Now at the point when the target pressure is equal to the wall pressure, the cell is said to be turgid. That is, full turgidity is attained when the target pressure equals to the wall pressure. At this point in time, the outward target pressure is exactly the same as the opposite inward wall pressure. The cell can stretch no further, and as a result, no more water molecules can get in. That state is said to be full turgidity. Now, plant cells do not burst because the tough cellulose within the cell wall that make up the cell wall, that is, can resist very high pressure. So as much as the target pressure acts outward, because of the presence of the cellulose, the cell wall will not burst, it will not give in to the outward pressure. Now, turgidity is important in plants in maintaining the shapes of the plant cells, thereby providing support and the upright posture of seedlings, leaves, and young stem is placed in distilled water or a very dilute solution, that is one that is hypotonic. Then water molecules will enter into the cell by osmosis and move into the cytoplasm and the subvacuole. As more and more water enters the cell by osmosis, the volumes of the cytoplasm and the subvacuole will increase, causing the cell to swell. So the cell swells as a result of the influx of water molecules, thereby expanding right from the subvacuole, the cytoplasm, and outward. The contents of the cell, that is the cytoplasm and the subvacuole, exert an outward pressure that presses against the cell wall. This outward pressure is known as the target pressure. The target pressure presses against the cell wall. Now, remember, the cell wall is rigid as a result of the cellulose. So, in resisting the stretching, the cell wall will exert an opposite and equal force. This opposite and equal force exerted by the cell wall is known as the wall pressure. So as the target pressure increases, the the wall pressure also increases because target pressure pressing against the cell wall is one that creates the opposite wall pressure. Now, as more and more water enters the cell, the target pressure will increase, and as a reaction, the wall pressure will also increase. So the cell will continue to expand, but remember, a point will be reached when this inelastic cell wall will resist further stretching. Now, at the point when the target pressure is equal to the wall pressure, the cell is said to be turgid. That is, full turgidity is attained when the target pressure equals to the wall pressure. At this point in time, the outward target pressure is exactly the same as the opposite inward wall pressure. The cell can stretch no further, and as a result, no more water molecules can get in. That state is said to be full turgidity. Now, plant cells do not burst because the tough cellulose within the cell wall that make up the cell wall, that is, can resist very high pressure. So as much as the target pressure acts outward, because of the presence of the cellulose, the cell wall will not burst, it will not give in to the outward pressure. Now, turgidity is important in plants in maintaining the shapes of the plant cells, thereby providing support and the upright posture of seedlings, leaves, and youngsters. In this lesson, we're going to look at water relations in plant cells, and specifically to demonstrate osmosis using plant tissues. Now, the plant tissue that we are going to use is derived from the potato tuber. This is an Irish potato tuber, which is basically a storage organ found in this particular plant. Now, using a scalpel, we make 3 cubes of about this size, and we bore out cavities at the center. So there'll be a hollow cavity at the center of each of the 3 cubes. That is, you can do that using a scalpel or a sharp knife to prepare a cube that is obtained from the potato tuber, and then we dig out this hollow cavity at the center. Then each of the 3 potato tuber is treated as follows. The first one is given the A treatment, in which salt crystals are placed at the center of the cavity, and the tuber is left unboiled, then placed in a petri dish that contains distilled water. The second tuber is given the B treatment, in which it is left unboiled and placed in a petri dish containing distilled water, but without any salt crystals at the center. And then the third tuber is given the C treatment, in which first it is boiled for about 5 to 6 minutes, and then salt crystals are placed at the center of the hollow cavity. Then the tuber is placed in a petri dish containing distilled water. The 3 tubers are left to stand for about 30 minutes, after which the following observations are made. In the first tuber, it will be observed that water will accumulate in the cavity. and the level of the water in the petri dish is going to go down. In the second tuber, we will see that no solution is formed in the cavity, but the level of the water in the petri dish reduces slightly. While in the third cube, we'll see that the level of the water in the petri dish remains the same, and the salt crystals remain unchanged, that is, they do not go into solution. Now, explanation of these observations. Now you can see that in the first potato tuber, the salt here in the cavity dissolves in the thin film of water found on the potato tissue, thereby forming a concentrated solution here at the center. This concentrated solution is hypertonic to the potato cells lining the cavity. So what happens is that water moves by osmosis from these cells lining the cavity into the cavity, thereby increasing the level of the solution there. The cells lining the cavity become hypertonic to the inner cells. Thus, water moves by osmosis from the inner cells to the cells lining the cavity, and this goes on until the outermost cells draw in water from the petri dish by osmosis, leading to a drop in the water level of the petri dish. So that's why you see after 30 minutes, the solution level in the cavity increases because water is drawn from the cells in the potato tuber by osmosis, and in turn they draw water from the petri dish by osmosis to replace the amount that is lost to the cavity, leading to a drop in the level of water in the petri dish. In the second tuber, we see that no solution forms in the cavity, but the water level reduces slightly in the petri dish. Now, how is that? This comes about because the cells in the potato tuber are hypertonic to the distilled water in the petri dish. So, by osmosis, water molecules are drawn from the petri dish and into the potato tuber. And this is what brings about a slight drop in the level of the water in the petri dish. In the third tuber, where there is no observable change, comes about because boiling destroys the cell membrane. Now, with the destruction of the cell membrane, no osmosis can take place. So the salt crystals remain undissolved. Water in the petri dish cannot be taken up by osmosis because of the destruction of the cell membranes of the cells in the potato tuber. So consequently, no water is taken up into the tuber, and there is no dissolution of salts crystals in the potato tuber. To this lesson. In this lesson, we look at active transport as one of the processes by which materials move into and out of living cells. Active transport is defined as the movement of substances from a region of low concentration to a region of high concentration across the cell membrane. The substances are moving from where they are lowly concentrated to where they are highly concentrated, and this process therefore occurs across
as cell membrane, and the substances tend to move against a concentration gradient, because the substances are moving from where they are lowly concentrated to where they are highly concentrated. It's like they're moving uphill. Now, this type of movement requires energy. Energy is required for the process of active transport to take place, because the substances are moving from a region of low energy area, that is low concentration, to a region of high energy area. So this movement is against a gradient, and for it to take place, energy must be available. It is for this reason why active transport can only take place in a living system where energy is generated through respiration, and this energy is then used in the movement of substances across the cell membrane. Now, the mechanism of active transport and exactly how it takes place is thought to come about in the following manner. Within the cell membrane, there are specialized protein carriers. These are protein substances whose function is to combine with substances that are being transported and then ferry them from one surface to the other. So like in this particular case, the substance to be transported attaches itself onto the protein carrier. Then the protein carrier moves across the cell membrane and deposits the substance on the other side. This movement of protein carrier molecules back and forth the cell membrane is what consumes energy. And this is the part of the process that requires energy. So by moving substances from one end or one surface of the membrane to the other, the cells can actively take up substances from their surroundings and into the cell, or remove unwanted substances or products from the inside of the cell and to the surrounding. In this chapter, we look at nutrition in living organisms. We can start by defining what nutrition is, and this is defined as the process by which living organisms obtain and utilize food substances or nutrients. Now, what is a food substance? Anything that is either taken into the body or is made inside the body from which the organism obtains nutrients is known as a food substance. Nutrients, on the other hand, is any substance that is a source of energy and structural components to a living organism. So from nutrients, living organisms obtain energy and substances that are used to make up structural components such as the cell membrane, organelle membranes, enzymes, and so on. So nutrition is a process by which living organisms obtain food substances. They can either make the food or obtain it from outside. Then from this food, they get their nutrients. And within the cells, the nutrients can be broken down to provide energy that drives many processes, or the nutrients can be used to make up components of various parts of the cell. Energy is put to a number of uses in a living organisms. For example, this energy may be used in the synthesis of cellular materials for growth and repair.
The energy may also be used in active transport of substances, and it can also be used to provide locomotion in organisms such as animals. Structural components, on the other hand, are derived from amino acids, which are types of nutrients, fatty acids, and mineral salts. These molecules make up the cellular structures, for example, the cell and organelle membranes, the cell wall in plants, enzymes, and hormones. So all living organisms must acquire food to obtain nutrients, and then from these nutrients they get their energy for various processes, and also to obtain their molecules that are used as structural components of various parts of the cell and therefore the organism. Now, there are two different types of nutrition known as modes of nutrition. There are two modes of nutrition, and these are: one, there is Autotroph Ism, and two, Heterotroph Ism. You can start by distinguishing the two. Autotroph Ism refers to the synthesis of food substances within the body of an organism. These food substances are made from simple substances using energy from an external source. Such organisms that are capable of making their own food are known as autotrophs. Example, green plants. While heterotrophism, on the other hand, is a mode of nutrition in which an organism takes in already made complex food materials from another organism. Such organism is said to be a heterotroph. So heterotrophs are organisms that obtain food substances that has been made by another organism. So they obtain their food materials from outside their body. They cannot make their own food, so they have an alternative source of food. That's why they refer to as heterotrophs. In this chapter, we look at nutrition in living organisms. We can start by defining what nutrition is, and this is defined as the process by which living organisms obtain and utilize food substances or nutrients. Now, what is a food substance? Anything that is either taken into the body or is made inside the body from which the organism obtains nutrients is known as a food substance. Nutrients, on the other hand, is any substance that is a source of energy and structural components to a living organism. So from nutrients, living organisms obtain energy and substances that are used to make up structural components such as the cell membrane, organelle membranes, enzymes, and so on. So nutrition is a process by which living organisms obtain food substances. They can either make the food or obtain it from outside. Then from this food they get their nutrients, and within the cells the nutrients can be broken down to provide energy that drives many processes, or the nutrients can be used to make up components of various parts of the cell. Energy is put to a number of uses in a living organisms. For example, this energy may be used in the synthesis of cellular materials for growth and repair. The energy may also be used in active transport of substances, and it can also be used to provide locomotion in organisms such as animals. Structural components, on the other hand, are derived from amino acids, which are types of nutrients, fatty acids, and mineral salts. These molecules make up the cellular structures, for example, the cell and organelle membranes, the cell wall in plants, enzymes, and hormones. So all living organisms must acquire food to obtain nutrients, and then from these nutrients they get their energy for various processes, and also to obtain their molecules that are used as structural components of various parts of the cell and therefore the organism. Now, there are two different types of nutrition known as modes of nutrition. There are two modes of nutrition, and these are: one, there is autotrophism, and two, heterotrophism. You can start by distinguishing the two. Autotrophism refers to the synthesis of food substances within the body of an organism. These food substances are made from simple substances using energy from an external source. Such organisms that are capable of making their own food are known as autotrophs. Example, green plants. While heterotrophism, on the other hand, is a mode of nutrition in which an organism takes in already made complex food materials from another organism. Such organism is said to be a heterotroph. So heterotrophs are organisms that obtain food substances that has been made by another organism. So they obtain their food materials from outside their body. They cannot make their own food, so they have an alternative source of food. That's why they refer to as heterotrophs. Two types of autotrophism. There are two main types of autotrophism depending on the source of energy, that is the nature of the energy that is used in the process. Because remember, in autotrophism, an organism makes its own food using energy. So based on the type of the energy that is used, there are two types of autotrophism, and these are namely chemosynthesis. Now this, as the name suggests, refers to the synthesis of complex food substances from simple inorganic substances using energy derived from chemical processes. So this is by use of chemical energy. Chemical energy that is derived from chemical processes. Chemosynthesis is mainly carried out by certain types of bacteria that live both in the soil and in water environment. Now we are not going to talk anymore about chemosynthesis because our main focus is on the second type of autotrophism, and that is photosynthesis. Now, just to contrast the two, and distinguish them. Photosynthesis is the synthesis of complex food substances from carbon dioxide and water using light energy. So in photosynthesis, the type of energy used is light energy, whereas in chemosynthesis, the energy that is used is chemical. Now, during photosynthesis, oxygen is given out as a byproduct and may be released to the surroundings. Photosynthesis is carried out by green plants. Mainly, these are the most common photosynthetic organisms. Algae also do carry out photosynthesis, and certain types of bacteria that are photosynthetic. Now, for photosynthesis to take place, light must be absorbed. That is, light energy must be absorbed. And most of the organisms that carry out photosynthesis have the pigment chlorophyll molecule. Chlorophyll molecule is found in green plants, green algae, green bacteria, and it is used to absorb light energy from the sun. So all plants that are green in color are so because of the presence of chlorophyll that absorbs light energy. Now, photosynthesis is a very important process in nature. Photosynthesis as a process is very important to the survival of living organisms. So you can look at the importance of this process in nature. So the importance of photosynthesis in nature. Now, the first one is that through this process, complex organic substances are formed and which act as food. So it's a source of food to living organisms, both to the photosynthetic organisms and the other living organisms that depend on it. The second important point for photosynthesis is that it enables the removal of carbon dioxide. Removal of carbon dioxide that is released by respiring living organisms during respiration from the atmosphere. Accumulation of carbon dioxide in the atmosphere is dangerous to the environment, for it will affect climatic conditions. So, through photosynthesis, the carbon dioxide produced during respiration in living organisms is removed, and this prevents the accumulation of this gas in the atmosphere, which may lead to global warming as one of the serious effects of higher than normal levels of carbon dioxide. The third point of importance is that through photosynthesis, carbon dioxide that is used in the process and food is produced. Remember earlier on we had said that apart from the food substance that is formed, oxygen is also produced. So through photosynthesis, oxygen is replenished into the atmosphere, and this can be used by living organisms during respiration to break down food substances to provide energy. The energy is used for many processes within the cell. So apart from producing the food, photosynthesis is very important in that it gets rid of carbon dioxide that is released by living organisms and also replenishes the oxygen that is used by the very same living organisms during is the main organ for photosynthesis. It has specific features that enhance the process of photosynthesis. Now we look at these features and how they enable the leaf as an organ to carry out photosynthesis efficiently. We first start by looking at the external features. Now for our purpose, we look at dicotyledonous leaf. But whatever features that are present and true for dicotyledonous leaf are also present and true for a monocotyledonous leaf. Now if you look at the external features of a leaf, there are those characteristics that enhance the process of photosynthesis. The first one is the color. Most plants have green leaves. The greenness is due to the presence of the pigment chlorophyll. Chlorophyll enables the plant to trap light energy, which is then used in the process of photosynthesis. The lamina of the leaf is quite thin. Most plants have thin lamella. This is to reduce the distance that light penetrates in order to get to the photosynthetic tissues. It also reduces the distance that carbon dioxide from the atmosphere must diffuse to get to the photosynthetic tissues. The leaf blade is also broad. This is to provide a large surface area for absorption of light and carbon dioxide. There is the petiole, which provides support by attaching the leaf to the stem, and the attachment of the petiole to the stem is such that it is at an angle. It supports the leaf, that is the leaf, at an angle for maximum light absorption. That is, so that the leaf is exposed in such a way that maximum area for light absorption is effected. So the petiole supports the leaf in the best possible angle for maximum sunlight absorption. There are veins in the midrib. The midrib and branching from the midrib are the veins. If this was a monocotyledonous leaf, then there will be no branching from the midrib. Instead, there will be many ribs that are parallel to the one at the center. That's making the parallel venation that is characteristic of the monocotyledonous leaves. But in this particular case, you have the midrib and the veins. These one provide support so that the leaf is exposed to maximum light absorption. Secondly, the midrib and the vein do contain vascular tissues that conduct water from the roots to the leaves and carry away the products of photosynthesis from the leaf to the other parts of the plant. So externally, those are some of the features that adapt the leaf to the process of photosynthesis. Now internally, the leaf has the following features, adaptive features for photosynthesis. One, the cuticle. Most terrestrial plants have a cuticle. This can be on the upper and also lower surface, or just on the upper surface only. The cuticle is transparent, it is non-cellular, waxy and waterproof. The transparency allows for the penetration of light to the photosynthetic tissues beneath. So light can penetrate through the transparent cuticle to get to the photosynthetic tissues. Being waxy and waterproof, it also reduces excessive loss of water by evaporation. And also being non-cellular, it protects the underlying tissues from mechanical damage and infection. Then the outermost tissue covering the leaf is the epidermis. There is the upper epidermis and the lower epidermis. The epidermal cells lack chloroplast, hence they are also transparent to allow light through to the photosynthetic tissues. In some regions, the epidermal cells are modified into guard cells. They are modified into guard cells. So, like here on the lower epidermis, you have these guard cells. That's a guard cell. That's another guard cell. Now, these guard cells control an opening. There is a pore between them, the stoma. That is the stoma. So the guard cells control the opening and closing of this stoma through which carbon dioxide enters the leaf to be used in photosynthesis. These guard cells also have chloroplasts in them, hence they are also photosynthetic. It is through these stomata on the epidermis that carbon dioxide from the atmosphere diffuses into, so carbon dioxide from the atmosphere diffuses into the leaf, while oxygen, that is a product of photosynthesis, diffuses out of the leaf. In most plants, the stomata are mainly located on the lower epidermis in order to minimize water loss. So you'll have most of the stomata on the lower epidermis, though also quite a number of plants, there are just as many stomata on the upper epidermis as there are low. It all depends on the environmental condition and how the plant is adapted to survive in its natural habitat. Another internal adaptive feature of the leaf is the mesophyll tissue. Now there are two types of mesophyll. Palisade Mesophyll beneath the upper epidermis and the spongy mesophyll below the Palisade Mesophyll. The Palisade Mesophyll consists of cells that are packed with numerous chloroplasts. They have numerous chloroplasts in them. So this green body, these are the chloroplasts. The chloroplasts contain chlorophyll molecules which absorb light energy that is used in photosynthesis. These palisade cells are located beneath the upper epidermis in order to receive maximum sunlight. So their location here ensures that they receive maximum sunlight. The cells are arranged such that their long axes are perpendicular. So this, if you see the orientation of the cells, this is their long axis. This is the long axis. It is perpendicular to the surface of this leaf. So that is the surface of the leaf, and the long axis of the cell is perpendicular to the leaf surface so that it can receive maximum light. So light will have to cross through a very long portion or part of the cell, and in the process most of this light would be absorbed. If it was not for this arrangement, if it was not for this orientation such that the long axis is perpendicular to the surface, then this will be the arrangement, and light will be crossing through a relatively smaller volume or area of the leaf, thus less light will be absorbed. But with this arrangement whereby the long axis is perpendicular to the surface, then this ensures that there is maximum light absorption. Another adaptive feature of the Palisade Mesophyll is that the cells are tightly packed so as to trap most of the incoming light. In some plants, the Palisade Mesophyll consists of several layers of cell in order to enhance light absorption, especially those plants that grow in areas that have minimum or very little light, like on the floor of forests or underwater. Such plants will have several layers of Palisade Mesophyll in order to enhance absorption of light. Then beneath the Palisade Mesophyll, there is a spongy mesophyll. The spongy mesophyll consists of cells that are irregularly shaped and are loosely arranged, thus creating air spaces between them. These air spaces promote diffusion of gases through the leaf. It enhances the diffusion of carbon dioxide from the atmosphere into the leaf, and also the diffusion of oxygen produced from photosynthesis out of the leaf. These large spaces also expose large surface area. These large surface area are exposed for gaseous exchange. So generally, the air spaces increase the surface area for gaseous exchange within the leaf. Now, the spongy mesophyll cells also do have chloroplasts, but they're not as numerous as in the Palisade cells. Within the leaf, we also have the vascular bundle. The vascular bundle consists of xylem and phloem tissues. The xylem conducts water and mineral salts from the roots to the leaf, where they are used in photosynthesis, while the phloem conducts products of photosynthesis such as sugar away from the leaf to the rest of the plant. So these are the adaptive features of the leaf, and each feature ensures or plays a role in the process of photosynthesis. So most leaves have these adaptive features in them to enhance. The chloroplast is one of the highly specialized plant cell organelle. It is organelle for photosynthesis. It has structural features which facilitate this process of photosynthesis, and some of these adaptive features include the following. 1. The chloroplast has numerous chlorophyll molecules, that is the green coloring pigment, whose function is to trap light energy. Secondly, the chloroplast has many internal membranes. These membranes are at some point stacked together to form regions known as grana, that is plural, and granum, singular. It is within these grana that chlorophyll molecules are contained. So the function of the grana is to provide a large surface area upon which chlorophyll molecules are contained and thus exposed for maximum light energy absorption. Within the chloroplast, we also have an aqueous matrix, an aqueous or watery medium known as the stroma. The stroma contains enzymes that speed up the process of photosynthesis. This stroma, being aqueous, it also provides the medium for photosynthetic reactions. And of course, the stroma is transparent. This is to allow light to pass through to the granum where chlorophyll molecules are contained. The oval shape of the chloroplast also does present a large surface area for absorption of light energy and also carbon dioxide from the surrounding within the cell. So in those ways, chloroplast is suited for the process of photosynthesis. Those features enable it to carry out photosynthesis. Photosynthesis involves a series of complex chemical reactions in which carbon dioxide and water react in the presence of light energy and chlorophyll to form carbohydrates. Oxygen is given off as a byproduct. Now the reactions can be summarized in the following equations. First, looking at the word equation, where carbon dioxide reacts with water in the presence of sunlight energy, which is absorbed by chlorophyll, to form carbohydrates in the form of sugar, with oxygen being given off as a byproduct. Now the byproduct, meaning that it is not the main reason for the reaction. The main product of photosynthesis is the carbohydrate. This can also be represented using chemical symbols, where 6 molecules of carbon dioxide combine with 6 molecules of water to give 1 molecule of carbohydrate, and 6 molecules of oxygen are given off. The process can be outlined in 2 general stages. In other words, this process of photosynthesis can be divided into 2 main stages, namely there's a light stage and the dark stage. The light stage is also known as light dependent stage because it requires light, the presence of light, for it to proceed. This light stage involves 2 processes. The 2 processes are 1, photolysis of water, and 2, synthesis of ATP molecules, that is the adenosine triphosphate molecules. In the first process, that is photolysis of water, it occurs in the granum. This process occurs in the granum where chlorophyll molecules are present. The chlorophyll molecules absorb light energy, and this energy is used in splitting of water into oxygen and hydrogen ions. In the process, some energy is also released. Chlorophyll absorbs light energy. The light energy is used to split water into hydrogen ions and oxygen. Now, during this splitting, some energy is released, and this energy is what is used to drive the second process that takes place during the light stage, and that is the synthesis of ATP molecules. So some of the light energy is used to combine adenosine diphosphate and a phosphate molecule to form the adenosine triphosphate, or ATP. The ATP molecule is the energy molecule in cells. Chemical energy is stored within ATP molecules. So when the energy is required, the ATP molecule will be broken down to its constituent adenosine diphosphate and phosphate, and the energy released in the process is used to drive a number of biochemical reactions in the cell. So that is the first stage, or the first part of photosynthesis, the light stage. It occurs in the granum part of the chloroplast. This is where we have chlorophyll molecules. Now, since this reaction requires light energy, chlorophyll must be present to absorb this light energy, and then the light energy is used to split water, and also is converted into chemical energy, and it is used to synthesize ATP molecules. In the second stage, known as the dark stage, this stage, since does not require the presence of light, is therefore referred to as a light independent stage. And during this stage, carbon 4 oxide combines with hydrogen to form simple carbohydrate. This combination, the reaction between carbon 4 oxide and hydrogen, is known as carbon 4 oxide fixation process. So the process that takes place in the dark stage is the carbon 4 oxide fixation, in which hydrogen, this remember is coming from the granum after the splitting of water, combines with carbon 4 oxide, most of which comes from the surrounding, for example in the atmosphere, to form simple carbohydrate. This process occurs in the stroma part of the chloroplast, because it is in the stroma that the enzymes that drive the process are found. Apart from being enzyme controlled, this process is energy consuming, and the energy is provided by the ATP molecules produced in the light stage. So this ATP that was formed during the light stage provides the energy that drives the carbon 4 oxide fixation process. The product of this fixation process are simple carbohydrates. Now, this simple carbohydrate is then built up into a number of molecules. So the simple carbohydrate that is first formed may be converted to glucose. This glucose can then be converted to fructose, and glucose and fructose together used to form sucrose. The glucose can also be converted into starch for storage. That glucose can be used to form cellulose, which makes up the cell wall of plant cells. Or alternatively, the simple carbohydrate may be converted into amino acids. Amino acids are the building blocks of proteins. Alternatively, the simple carbohydrates can be used to form fatty acids and glycerol. Fatty acids and glycerols are used to make up lipids. So, after formation, the first carbohydrate that is formed will be glucose. Or, again, depending on the needs of the plant, the simple carbohydrate can be converted to amino acids for protein synthesis, or fatty acids and glycerol that are used to make lipids for the plant. In this lesson, we look at factors influencing the rate of photosynthesis. The rate of photosynthesis is affected by a number of factors. These factors include light intensity, carbon dioxide concentration in the atmosphere, temperature, and water. It is the level of each of these factors that will determine the rate at which photosynthesis will take place. Now, all these factors must be present at their optimum levels. So talk about the optimum levels for photosynthesis to proceed at the fastest rate. Should any of these factors be at their lowest point, then the rate of photosynthesis will be low regardless of the level of the other factors. For example, if for instance light intensity is at its lowest level, then the rate of photosynthesis will be slow regardless of the concentration of carbon dioxide in the atmosphere, regardless of the surrounding temperature, and regardless of the availability of water. In the same case, if carbon dioxide concentration is very low, then even if you have enough light intensity, the temperature is optimum, there is water available, the rate of photosynthesis will be low. Now, the factor at its lowest level is called the limiting factor. That factor, which at a given time is at its lowest level, be it light intensity, be it carbon dioxide concentration, the temperature, or water availability, will be the limiting factor. So, for example, under low light intensity, the rate of photosynthesis will be low regardless of how high the levels of carbon dioxide, the temperature, or water availability are. The light, in this case, will therefore be the limiting factor, and the rate can only increase if the light intensity is also increased. Now, we look at the effect of each one of these factors on the rate of photosynthesis, starting with light intensity. Now, as light intensity increases, the rate of photosynthesis also increases up to a point and then levels off. This is illustrated in the graph. So if we start at a particular level of light intensity and we increase the intensity, the rate of photosynthesis is going to increase correspondingly up to a certain point, beyond which it levels off. Now, you know the role of light. Recall the role of light in photosynthesis. Light provides the energy for photolysis of water and also energy for carbon dioxide fixation. So as we increase the light intensity, there is more energy available for photolysis and carbon dioxide fixation. But a point is reached beyond which, even if we raise the light intensity further, the rate of photosynthesis levels of it remains constant. So if you look at the graph, that point that produces the highest rate of photosynthesis corresponds to the optimum range of light intensity. A further increase beyond the optimum range will not yield any increase in the rate of photosynthesis. Why? Because the other factors such as carbon dioxide and temperature, which are required for photosynthesis to proceed, are not increasing, and thus they become limiting. So beyond this optimum range of light intensity, the other factors, carbon dioxide and temperature, become limiting because they are not increasing in step with the light intensity. So when you start at a very low light intensity, light was the limiting factor. But as you increase the intensity, the rate will also increase up to a certain point. That is the optimum range. A further increase beyond the optimum range will have no effect on the rate, because beyond this point, it is not light intensity that is limiting. It is carbon dioxide concentration and temperature that become limiting. Now, turning to the second one, the second factor, that is carbon dioxide concentration. See, under normal conditions, the concentration of carbon dioxide in the atmosphere is about 0.04%. There may be occasional small fluctuations, either rising above or below the 0.04%. Now, despite these occasional fluctuations, the level in the atmosphere remains remarkably constant. It's always at about this 0.04%. This normally low atmospheric carbon dioxide concentration is a major limiting factor to photosynthesis. So under natural conditions, carbon dioxide level is what limits the rate at which photosynthesis will take place. Now under artificial conditions, the levels may be increased, and thence the rate of photosynthesis will also increase. Now we can look at the relationship between carbon dioxide and the rate of photosynthesis in the graph below. So starting at low carbon dioxide concentration, so as we increase the carbon dioxide concentration, the rate also increases, but up to a certain point. When we attain the highest rate, that is the highest rate. So the level that produces the fastest rate of photosynthesis is said to be the optimum range. Now, if we continue increasing, that is under experimental conditions, if we continue increasing the level of carbon dioxide beyond the optimum, the rate of photosynthesis will remain constant. Why? Again, here the reason is the other factors that are necessary for photosynthesis to take place have remained constant. So increase in carbon dioxide alone beyond its optimum range under those prevailing conditions will have no impact on the rate of photosynthesis. So the rate will proceed at the optimum range of CO2. It is as if the extra carbon dioxide available have no role to play in photosynthesis, because remember, the other factors are not increasing in step, so they become limiting. Those factors that are at their lowest concentrations or levels become limiting. So even if we increase carbon dioxide beyond a certain point, the level becomes constant. It remains constant rather with photosynthesis, because the other factors, temperature and light, are now limiting. The third factor is temperature. The light-dependent stage of photosynthesis is not affected by temperature. Remember, there are two stages of photosynthesis. There's the light stage and the dark stage, also known as light independent. So light stage does not depend on temperature, is not affected by the temperature, but the dark stage is influenced by temperature. This is because the reactions that take place in the dark state, that is the carbon 4 oxide fixation, is enzyme controlled, and enzymes are affected by temperature. Hence, an increase in temperature leads to an increase in the rate of photosynthesis because the temperature affects the second stage. So as the temperature rises, such a low point here, an increase in temperature will also lead to an increase in the rate of photosynthesis up to a certain point. Beyond that point, the point that produces the highest rate, that will be the optimum temperature range. Now, beyond the optimum temperature range, a further increase results in a decrease in the rate of photosynthesis. Now, in most cases, the optimum range of temperature is usually between 35 and 40 degrees C in most plants. So, why does the rate decrease beyond the optimum? It's because as the temperature rises, the enzymes become more activated, so they speed up the rate of photosynthesis. But when the temperatures rise beyond the optimum, the enzymes get destroyed. We say enzymes become denatured. And as more and more become denatured, the rate decreases until it may eventually come to a stop. Now, water as a factor affects many reactions in a plant, not just photosynthesis. And even photosynthesis, water is involved in a number of ways as a raw material, and therefore if it is deficient, then the rate will go down. Water is also necessary to make the cells turgid, thereby providing support for the leaves to open and spread out, thereby providing a large surface area for absorption of light, and also the turgidity makes the stomata to open to allow carbon 4 oxide to diffuse into the leaf. Now, since water has so many functions in a plant, it is impossible to directly relate its availability and the rate of photosynthesis like these other two factors. So we just note that water is necessary for photosynthesis to take place. Without water, the process will not proceed. And the lower the water availability, the lower the rate of photosynthesis. Limit the process of photosynthesis. Now, a limiting factor is any factor that prevents or limits the increase in the rate of a reaction when all the other factors are at optimum levels. Photosynthesis, like many other processes, are affected by several factors, and when any one of these factors is at its lowest level, then it's going to limit the rate at which photosynthesis will be taking place. In photosynthesis, light intensity, carbon 4 oxide concentration, and temperature interact in influencing the rate of photosynthesis at any one given time. So these three factors are very, very critical in determining the rate of photosynthesis. Now, for example, if any of these factors is increasing at any one particular time, then the rate of photosynthesis will also increase until a certain point, then begin to level off. For example, increasing carbon 4 oxide concentration at a constant temperature of 25 degrees C will increase the rate of photosynthesis at optimum light intensity. For example, if in this graph here, as you increase the light intensity, the rate is going to level off at this optimum. So let's call this is the optimum light. That is the optimum light intensity. Now, at 0.03% carbon dioxide, the fastest rate of photosynthesis is attained at that point. Now, if we increase the CO2 concentration from 0.03 to 0.1%, because remember, in the atmosphere, the normal concentration of CO2 is about 0.04%. Now, if we increase it from 0.03 to 0.1, then the rate of photosynthesis at this optimum light intensity rises, so that we have a higher rate of photosynthesis. So at a constant temperature of 25 degrees C, if we vary the CO2 level, if we increase the CO2 level, as long as light intensity is not limiting, that is, the light intensity is at its optimum, then we expect the rate of photosynthesis to also increase, because at 0.03% and with light intensity at its optimum and temperature at 25 degrees C, then this low 0.03% carbon dioxide will limit the rate of photosynthesis to this point here. So the only way to increase the rate of photosynthesis under such conditions is to increase the CO2 level from 0.03 to 0.1%. Similarly, if the temperature were to be raised, in the same setup, we have to raise the temperature from 25 degrees C to 30, the rate of photosynthesis will also be higher. You can illustrate this in the graph. Now here, for comparison, this was the rate of photosynthesis, that is in curve 1, at optimum. Again, this is our optimum light intensity. Optimum is that point when the level flattens off. Now, for comparison, when the temperatures were 20 degrees C and the CO2 level was at 0.03%, this is the fastest rate of photosynthesis attained. If we increased the CO2 level and also increase the temperature, then the rate of photosynthesis increases substantially to this point. And if you are to increase the CO2 level further, but at the same temperature, the rate will also increase to a higher point. So you can say that in curve 2, the temperature is not limiting, but compared to curve 1, the temperature being higher, it produces a higher rate of photosynthesis. CO2 level is also higher, so this will together contribute to a higher rate of photosynthesis. Now in curve 3, when the CO2 is increased further from 0.01 to 0.1 and the temperature is constant, the rate will also increase. So we can see that by increasing the level of the factor that is at its lowest point, we will influence the photosynthesis process by also increasing its rate. So remember, photosynthesis requires optimum level of the factors that are involved in the process. That factor, which is at its lowest point, will be limiting the process. So the only way to increase the rate of photosynthesis at any one given time is to increase the level of that factor whose magnitude is at its lowest point. Like in this particular case, in curve 1, you can see the rate is limited because of the low carbon dioxide and relatively low temperature. By increasing the temperature and also increasing the CO2, the rate increases substantially in curve 2. In curve 3, by increasing the CO2 level, the rate will also increase substantially. Now from this we can project and conclude that if you were to increase, say if you were to increase the temperature slightly higher to say 35 degrees C, then we would expect the graph to increase and reach the optimum level at a higher point, because the higher temperature will activate the enzymes more, and the process, especially the dark stage reaction, will proceed faster. But of course there's a limit as to how high we can increase the temperature, because beyond 35 then the temperatures will be too high and enzymes that drive the dark stage reactions, that is the carbon dioxide fixation, would be denatured. So there's a limit to which we can increase the temperature. Also there's a limit to which you can increase the carbon dioxide, because under natural conditions the levels of CO2 in the atmosphere is normally fairly low, and if we increase the levels to too high, again it's going to have negative effect on the environment. For example, high CO2 level in the atmosphere may lead to a greenhouse effect, that is retention of heat in the atmosphere, producing devastating effects in the environment, in the climatic conditions, and generally negatively affecting living organisms. But as a rule, under normal conditions, if we increase the factor that is at its lowest level, then we'll also... In this lesson, we want to show that photosynthesis has occurred in a plant. Now, one of the most convenient way to show that photosynthesis has taken place is to test for the presence of starch in a leaf. Why starch? Because during photosynthesis, carbohydrates are formed and excess glucose is converted into starch that is then stored in starch granules within the chloroplast. Now, how do we make sure that the starch that has been formed through photosynthesis and not from the storage regions? In other words, how do we ensure that the starch that is present in the leaf has been formed through photosynthesis and it's not as a result of carbohydrates being transported or translocated from other parts of the plant to the leaf? Now, the way to ensure that the starch is just from photosynthesis and not any other source is by destarching the plant. So we destarch, and destarching involves placing the plant, and conveniently to be a potted plant, in darkness for a period between 36 to 48 hours. And during this time, all the starch in the leaf will be hydrolyzed, and either the glucose resulting be used in respiration or can be formed into fructose and sucrose and then translocated to other parts of the plant. So the first step is to destarch the plant. Once destarching has taken place, the plant is then exposed to sunlight for a period of 4 to 6 hours, also making sure that all the other factors that are necessary for photosynthesis, like water, temperature, carbon dioxide, are present and optimum. Once that has taken place, then we should expect that photosynthesis will have occurred in the plant, and the excess glucose will be converted into starch. Now, the following are the steps in testing for starch in a leaf. The first one, you obtain the leaf from the plant that has been exposed to sunlight for a period of about 4 hours. Then place it in boiling water. You place it in boiling water. This is to kill the protoplasm, that is to kill the living part, and also stop the process of photosynthesis. In addition, placement in boiling water also helps to rupture the starch grains. The starch granules are ruptured, and that will make it easy to test for their presence. Then, in the next step, the leaf is placed in either a beaker or a boiling tube containing ethanol. Ethanol is an organic solvent, one that is commonly used to extract chlorophyll molecules from the leaf. After placing the leaf in a beaker or boiling tube containing ethanol, the beaker with the ethanol and the leaf is then transferred to a larger beaker containing hot water. So the boiling tube or the beaker containing the leaf and the ethanol is placed in a water bath, because we do not heat ethanol directly because it is highly inflammable. So we heat the ethanol indirectly in a hot water bath. The heating lasts for about 5 minutes, and during this time, chlorophyll molecules will be extracted from the leaf. Now, the extraction is important because when testing for starch, the presence of chlorophyll molecules would obscure a positive test. So it is important that you remove the coloring pigment, that is the chlorophyll molecules, from the leaf. After about 5 minutes of heating ethanol, the leaf is then placed in a petri dish containing warm water, or it can also be placed in another beaker containing warm water, preferably warm water. This is to remove residual chlorophyll molecules and also ethanol. And warm water also prevents the leaf from drying and becoming hard and brittle. So after washing in warm water, then it is transferred into a beaker, into a petri dish, or it can be spread out on a white tile. Then you pour, you flood the leaf. You apply as much iodine solution as possible over the leaf, and if it turns to blue-black, then that will show that starch is present, and therefore can conclude that during the time that the plant and the leaf were exposed to sunlight, photosynthesis took place, carbohydrates were produced, and the excess converted into starch for storage. So that way we can conclude that photosynthesis has occurred due to the presence of starch in the leaf. We want to investigate the necessity of carbon 4 oxide in photosynthesis. Potassium hydroxide solution is used to absorb carbon 4 oxide in the conical flask that contains leaf A. Leaf B, is exposed to all the requirements of photosynthesis, including carbon 4 oxide. Before the fitting of the conical flask, the potted plant is kept in a dark place for a period of 48 hours so as to discharge it. To discharge is to remove all the starch from the plant, especially in the leaves. After the period of 48 hours, the plant is set up as indicated in the diagram. Then it is exposed to sunlight for a period of 2 to 3 hours. After exposure to sunlight, the leaves are detached and tested for the presence of starch. Leaf A will show a positive test. It will give a blue-black color. Remember to test for starch. The chlorophyll must be removed by boiling the leaf in ethanol, then pouring iodine solution on the leaf in a petri dish or on a white tile. Formation of the blue-black color shows the presence of starch. So in leaf A, starch is present because the leaf was exposed to all the requirements for photosynthesis, including carbon 4 oxide. Leaf B, the one that was in the conical flask, gives a negative test for starch, forming the brown color. This brown color is the color of iodine solution. Showing the absence of starch. So, leaf B lacks starch because the potassium hydroxide solution absorbed all the carbon 4 oxide that was present in the flask, such that the leaf could not carry out photosynthesis even though it was exposed to sunlight. So we can see that without carbon 4 oxide, even though sunlight is present, the temperature is optimum, no photosynthesis can take place. In this lesson, we are going to investigate the necessity of light in photosynthesis. We use a potted plant which is first discharged, called the discharging process. This is when the plant is kept in the dark for about 48 hours to ensure that all the starch in it is used up. Then, when it's exposed to conditions that can enable it to carry out photosynthesis, we test for the presence of starch, and if starch is present, then we'll conclude that photosynthesis will have taken place. So in this experiment, a potted plant is first discharged. Then 2 or more leaves are covered with a light-proof material such as aluminium foil. The plant is then transferred to an area where it will be exposed to light for about 3 hours. After 3 hours in sunlight, the leaves are detached and uncovered and then immediately tested for the presence of starch. So This is how the leaf would look like at the end of the experiment. You can see that the parts of the leaves that were uncovered, that is the exposed green parts, carried out photosynthesis and were able to form starch. So they presented the blue-black color. But the parts that were covered with aluminium foil show brown color, meaning the absence of starch. Now, why is this so? In these areas, no starch was formed because light was excluded. So without light, photosynthesis experiment, we want to investigate the necessity of chlorophyll in photosynthesis. This is achieved by the use of a variegated leaf. A variegated leaf is one that has some patches that lack chlorophyll, and therefore these patches have different colors, like it can be white, like in this particular case, or it can be yellow, cream, red. Basically, the absence of chlorophyll allows that part to have a different color other than the green. But the parts that have chlorophyll are green in color. So a leaf that has regions that are green in color and other regions that are non-green is said to be variegated, and it's the one that is used to show the necessity of chlorophyll in photosynthesis. Now, in this experiment, we just make sure that the plant with variegated leaves is exposed to light for at least 3 hours. This is to allow for photosynthesis to take place. Then we detach or remove a variegated leaf from the plant. Now, it would be good if we draw a large label diagram of the leaf before testing for starch to show the distribution of the chlorophyll pigment. The distribution of the chlorophyll pigment coincides with the green area. So the green area will indicate the presence of chlorophyll. Then we test the leaf for starch in the normal manner, that is, first dipping it in boiling water to stop photosynthesis and to rupture starch granules. Then we boil it in ethanol to extract the chlorophyll molecules. Then wash in warm water and spread on a tile, and then flood with iodine solution. So when we test for starch by flooding with iodine solution, this is what we see. The non-green area, in this particular case the white region, will give the brown color, showing absence of chlorophyll. But the green regions will give us the blue-black color, showing that starch is present. And for starch to be present, then photosynthesis must have taken place. Because remember, this plant was initially discharged and then exposed to sunlight in order for photosynthesis to take place. So the presence of starch in the green area shows that it's only where chlorophyll is present that starch is formed, and therefore it's only in this area that photosynthesis took place. Absence of starch in the non-green area shows that where there is no chlorophyll molecules, no photosynthesis took place, and therefore we can conclude that without photosynthesis, without chlorophyll, we investigate the gas produced during photosynthesis. As you recall, oxygen gas is a byproduct of photosynthesis, And this production of oxygen is easily collected from aquatic plants such as the pondweed, which is commonly referred to as the Elodia. Other plants that may be used include the Spirogyra and the water lily. Now, given that water usually has a low concentration of carbon dioxide, the water is enriched with CO2 through the addition of sodium hydrogen carbonate, which dissolves to release carbon dioxide. So the addition of sodium hydrogen carbonate boosts the amount of carbon dioxide in the water so as to promote the process of photosynthesis. Now we also make sure that there is adequate light, because light, just like carbon dioxide, are factors that are required in order for photosynthesis to take place. So the setup is put up like this diagram here, and then, as I mentioned, you make sure that it is exposed to sunlight to allow photosynthesis to take place. Now we leave the setup in the sunlight until sufficient gas will have collected in the boiling tube. Then we test the gas using a glowing splint. We test the gas using a glowing splint, and if the gas is oxygen, then the glowing splint will relight or will glow brighter. So from there, we will conclude that the gas produced during this experiment is oxygen. Now, this experiment can be used to investigate various factors on the rate of photosynthesis. In other words, we can use this setup to investigate various aspects of photosynthesis, and this includes the effect of light intensity on the rate of photosynthesis. By using a light source, so we modify the experiment slightly. So we provide a light source. This light source, instead of sunlight, we get light energy from this bulb. Now, since we do not want temperature to be a factor in the experiment, we do not want temperature to vary, there's a special heat screen that we use to prevent the heating up of the setup by the bulb. Otherwise, the setup remains just like in the first one. Here we have water that is enriched with sodium hydrogen carbonate. We use an aquatic plant like Elodia. Now we can vary light intensity by varying the distance between the lamp and the plant. The longer the distance, the lower the light intensity. And the shorter the distance, the higher the light intensity. So rate of photosynthesis can then be recorded and compared, because you can determine the rate by counting the number of oxygen gas bubbles being produced per unit time. For example, you can count the number of bubbles per minute at each distance. So let's say you can start, if the distance is 15 centimeters, we can count and record the number of oxygen bubbles produced per minute. Then we move the distance further to say 20 centimeters. Then we count and record the number of bubbles of oxygen produced per minute. So you keep varying the distance so that from the data we'll be able to relate the distance, which determines the light intensity, and the rate of photosynthesis measured by the number of oxygen bulbs produced per minute. So the same experiment can be used through modification to investigate the effect of carbon 4 oxide concentration on the rate of photosynthesis. For example, by using known quantities of sodium hydrogen carbonate, and therefore we know an estimation of the concentration of the CO2 in the water, we can then determine the rate at which oxygen is being produced by keeping the light intensity constant and also the temperature constant. So we can vary the concentration of the CO2 and then measure the rate by counting the number of oxygen bubbles being produced. We can also investigate the effect of temperature by placing this setup in different temperatures and counting the number of oxygen gas bubbles being produced at a given temperature. Then from here we can relate the effect of varying temperature on the rate of photosynthesis, just like we can relate the effect of different concentration of CO2 on the rate of photosynthesis. So this experiment is very important because we use it to measure or investigate the various factors and how they affect the rate of photosynthesis. The main factors that we use or that we investigate include: one is the light intensity, temperature, and the concentration of carbon 4 oxide.